Oh, hello there. My name is Joe Komsky, and I'm the Senior Director of Philanthropy at Sanford Burnham Prebys. Thank you so much for joining us today for our Insights lecture. Today's topic will be on addiction. Insights is our special lecture series where you get a chance to learn about our research and ask questions of our scientists. Since some of you are new to Sanford Burnham Prebys, I wanted to tell you a little bit about why it's such a special place. We conduct both foundational research, sometimes called basic research, and drug discovery. So what does that mean? Well, our foundational research is mapping the human biology of our bodies, of disease, so that we know how diseases interact with our bodies. Think of it a little bit like the Avengers. You've got Nick Fury, you've got the Agents of S.H.I.E.L.D. They're out there making sure everything in the universe is going well. But when it's not, when something starts to happen, when Thanos comes along, we need to recognize that something is wrong. And we have scientists who can go out and create medicines ourselves to fight the evil that's going on, right? To fight the bad things. And those things that we look at are cancer, heart disease, neurodegenerative diseases, many more. Now, the guys you're going to talk to today, they're like Iron Man and Thor and Captain America. These are our drug discovery folks. And what they're doing is they're looking for the weapons that will fight the diseases that are affecting our body, our universe. So today we're going to hear about research and drug discovery that lead to medicines that will help people fight addiction. Addiction is one of the primary health crises of our time, and it affects millions of Americans, and many of us know someone who fights this disease. Now, we want to make an important point here. We are not experts in the behavior or psychology of addiction, but we are experts, as we said, in the biology of the human body. Because of that, we can find medicines that can utilize our biology to lessen the pleasurable effects of addiction. And we feel that if we can do that, then this can work with behavioral and other treatments to give people a better chance to overcome their addictions. So first, a quick bit of housekeeping. We're going to give each scientist about 10 minutes to describe their work. And during the presentation, if you have questions, you can submit them via the Q&A button on the bottom of your screen. And we will present them to the panelists. First up is Dr. Nicholas Cosford, who has spent more than 25 years assembling small molecules into disease-fighting chemical compounds and has been with the Institute since 2005. Dr. Cosford is a professor and deputy director of the Sanford Burnham Prebys Cancer Center, one of only 7 National Cancer Institute designated basic research centers in the US. Nick has a distinguished record of drug development that includes more than 40 issued patents and more than 40 additional patent applications pending. His SBP research team focuses on cancer, neurological disorders, and infectious diseases. In addition to his pioneering addiction research, Nick also has recent breakthroughs in the development of drugs to treat deadly cancers. Take it away, Nick.
Thanks, Joe. So as Joe pointed out, at Sanford Burnham Prebys, we're really passionate about finding cures for disease, and top amongst our passions is to find treatments for mental health conditions. And if you go to the first slide, the focus of today's presentation is addiction. And of course, many of us are touched by addiction. We have family members, loved ones who struggle with this problem. It's a huge problem across the world and in the United States. And if you look at the numbers on this slide, they're really quite staggering. So in terms of, standard alcohol use and tobacco use, the numbers are huge. But in addition, there's so many illicit drugs available. And, this is a massive burden on society and the economy of treatment. And again, in addition to tobacco and alcohol, as many of you know, a massive problem is prescription pain relievers which often leads to the use of heroin. So if you go to the next slide, one of the big problems is that there are relatively few available medicines to treat addiction. So here we've just listed some of the drugs which can be taken. Interestingly, among these medications, all of them are really very old. The most recently approved drug was Chantix, which was approved in 2006. So it's been 14 years since there was a new medication to treat addiction. And interestingly, this is in part because pharmaceutical companies have not really spent a lot of time focusing on this area. And, you know, we can talk about perhaps why that is, but this is a real reason why we are particularly motivated to work around addiction and fill this gap that is this need, which is not being filled by pharma. So if we go to the next slide, I'm just going to talk a little bit about communication within cells, and in particular in the brain, to kind of set the scene for what my colleagues Michael Jackson and Doug Sheffler are going to talk about. And on the left, obviously, people communicate by speaking and hearing. Now, as you know, the body is made of millions and millions of cells, single cells, which all need to communicate with each other in order for us to function as human beings. In terms of brain cells, the majority of brain cells are called neurons. And these are interesting because they're actually very, very long cells. They stretch from the brain to various parts of the body and within the brain. So they communicate through electrical transmission. But the issue here is there has to be a way to control this communication, because if that wasn't the case, every neuron would be constantly firing and you would just seize up very quickly. So the way neurons control their communication is that they have these gaps between them. And this is what the video is showing. This gap is called a synaptic cleft. And these little purple things are chemical compounds which are released by one side of the neuron and activate what we call receptors on the adjacent neuron. These chemicals are called neurotransmitters, and these are critical with respect to how we treat problems in the brain like addiction. So if you go to the next slide, one thing we don't always realize is that the brain is an organ which takes more energy, takes and uses more energy in the body than any other organ. Of course, it's highly complex, and it consists of various sections, sort of like the equivalent of organs in your body. They have fairly complex names like the prefrontal cortex, basal ganglia, extended amygdala, and so on. But these are basically different parts of the brain which have different functions and work together. The prefrontal cortex, for example, is involved with anticipating things, being occupied by things. The basal ganglia are involved with binging and intoxication, and then the amygdala, withdrawal and negative effects of addiction. And so really these are three components of the brain that are really important, that are, there are problems in the way these parts communicate, which causes addiction. So later on, you'll hear a little bit more detail about some of these components and also some of the other parts of the brain, and in terms of how we're tackling this problem. Finally, for my section, before I hand on this slide, some of you may have already seen this type of thing, and this shows starting from the left and working to the right. The left is where we start to discover new medicines. So in the laboratory, we're working at Sanford Burnham Prebys. We have graduate students, postdocs, technicians, senior scientists who are working constantly to understand what's going on in these cells, what's going on in these parts of the brain that causes the problems of addiction. And once we've honed in on these key targets, this is a process called target identification, then we start to develop ways to experimentally test if we can find what we call small molecules, chemicals, that can either activate or inhibit these targets and produce the effect that we're looking for. So that's the assay component, and then we screen. You'll hear more about this momentarily from Michael Jackson. Then we move into what we call target validation. So at this point, we have to show that these compounds, these chemical compounds that we make that eventually could be drugs, we have to show that they are effective in doing what we want them to do, in this case, treating addiction, but also that they are entirely safe to be given to a human. So this sort of light blue zone on the left is where we focus our efforts at Sanford Burnham Prebys. Now, once we are successful and we move everything to the right, we come up to the green section, and at that point, what we do is we prepare all the information and documentation needed to present to the Food and Drug Administration, and then they will give the green light that we can proceed with human clinical trials. And so then that's phase 1 clinical trials, which only looks at the safety of a drug. So then you find healthy human volunteers and you just make sure that there's no problems with taking the drug. Phase 2 continues to look at safety, but then also efficacy. So is it going to work to treat people who are smokers, who have problems with drinking and so on? And then finally, phase 3 is a much bigger trial looking at efficacy and safety. And again, you may be even more familiar with this currently, given the interest in finding a treatment for COVID-19. So it's the same process that you go through. Ultimately, all these data are shown to the Food and Drug Administration, and then they will approve the drug. Now, just finally, the last couple of minutes. So again, to be tested in humans, as I said, drug must be safe and it must work. This zone on the left-hand side, the light blue, is in terms of how much it costs to do that, it's actually comparatively low. As the further you move to the right, the more expensive it becomes. And so you can see the target screen, that entire left-hand side, $1 to $4 million, that sort of ballpark, to get to something that may be ready to for the FDA to move into phase 1. Beyond that, you're looking at 12 to 15 million, up to tens of millions to move all the way through to approval. So at this point, I'm going to finish, and I'm going to hand over to my colleague, Michael Jackson.
Thanks, Nick. Next, I'd like to introduce Dr. Michael Jackson, who is the Senior Vice President of Drug Discovery and Development and leads the operation of the Prebys Center, a 60-person academic drug discovery enterprise embedded within the Institute. The mission of the center is to discover completely new classes of drugs directed to disease targets identified by investigators at the Institute and their collaborators. Through a 30-year career in both industry and academia, Dr. Jackson has experience in all aspects of translational research, with a focus on discovering and advancing first-in-class therapeutics to the clinic. Since joining Sanford Burnham Prebys in 2009, he has worked closely with senior leadership to seek new avenues of support for preclinical and clinical drug development to support our mission to translate basic research to new therapeutics and medicines. Michael, please go ahead.
All right, well, thank you very much. So what I would like to sort of follow up from Nick to talk about it, what is drug discovery? Tell you a little bit about the process and the nuts and bolts of how we go from an idea of what we want a drug to do to actually identify a physical substance that actually we can put into a pill and then take to the clinic. So those are the components I want to talk about. We are going to later, you're going to hear from Doug about 2 brand new potential medicines that we have come up with using this process. And ordinarily, this type of activity would be done traditionally in a pharmaceutical company. So if I have the next slide, basically the process where drugs normally would be expected to come from, most academic organizations, and many people would consider Sanford Burnham Prebys to be an academic organization and would associate us primarily with discovering the basis of disease and the components that have gone wrong, but not so much the drugs. Ordinarily, you would associate those sort of things with Merck or Pfizer or somebody. However, as Nick mentioned earlier, some diseases, for example, Alzheimer's disease, addiction, depression, these are diseases for which pharmaceutical companies have somewhat retracted their effort over the years. And so about 10 years ago or so, just a little bit more than that, the NIH made a major initiative to try and encourage academic organizations to become involved in this process of coming up with new drugs. And we encouraged by, as I say, not just the NIH, but also by disease advocacy groups and by philanthropists. They're like, well, you can't just sit there and say, let's not do anything. These are big issues for society. We have not had a single new drug for Alzheimer's disease, for example, since I think it's 2003. These are some diseases that really require attention: addiction, Alzheimer's. And so we were able to get some resources to assemble the capabilities to be able to do the early stages of drug discovery at Sanford Burnham Prebys. And we're very proud to be able to tell you some of the results of that a little later, particularly with these 2 new drugs we've got that are advancing for addiction. But let me tell you how this kind of works. So academia, and in fact, just about all drugs have their origination in academia through the identification of a drug target. Now, what we mean by the drug, that is the component of the human body that the drug is going to bind. And what I'm showing here on the left-hand side is a protein. And so in general, most of the targets of drugs are, which the drugs we're talking about here are primarily small molecule chemicals, little white powders, very small molecules, and shown in color there, you see it binding onto this structure. And this structure is the structure of a protein. And there are many different types of proteins. They do particular things. They can cleave things. They can add things. They're doers in cells. And we're going to identify the component in the human body that we want to modulate its activity. And we modulate it by binding these small molecule chemicals, and in so doing, either stop that protein doing its activity or activate it, or there's another way, modulate its activity. So you can kind of see from the get-go, a research scientist has this idea: I want to switch off this particular component. It could be a sensor in the human body. Sensors, same word as receptor, a sensor. You kind of want to switch it off or switch it on. And as Nick tells you, the whole of the transmission at the synapse is chemical in nature. So chemicals are released, they bind to a protein or a receptor, a sensor. And so we're looking for compounds and chemicals that could influence that particular binding. So that's the game plan. And those things we see as a sort of lock and key analogy. That little pocket that you see in the space filling model of the protein has got a very specific shape and scale and shape and size. And so we're looking for something that fits into that very precisely. Now, how are you going to find something like that? That's a problem, right? You can't just say, well, I think it should be one of these. What we do is to actually screen. And so the next slide shows you an example of many of different ball and stick diagrams of chemicals. And if you didn't follow your chemistry section at school, each of these is showing sort of carbon, oxygen, nitrogen, organic molecules. And we at Sanford Burnham have assembled a collection, a physical collection of these drug-like molecules. We have about 1 million of them. Right, well, that's good. But how are you going to find which of those million fit in the pocket? Well, if we did that one at a time, that would be 1 million experiments. That's a lot of work, to say the least. And we can't do that one test tube at a time as shown here. And so what has happened over the last 10 years, or maybe more than that, about 20 years, people have deployed, and we have deployed robotics. And so you can see down there on the left-hand side, a little orange arm of our robotic system, what's going on there. It's moving around 1,536 experiments at a time. And so it's each of those, from a test tube, it's been miniaturized into a tiny, tiny little well. And because it's so miniaturized, we can no longer move liquids by hand. So what you see is this little dot bouncing up from a pool there. That's actually using sound waves to ping out little tiny droplets of liquid into these little wells so that we can miniaturize each of the experiments, so that when we do 1 million experiments, we can bring the cost down and bring the experiment to be much more manageable to achieve our end goal, which is to find a chemical that specifically binds that pocket. And so once we've done that, that's the beginnings of a drug. That's kind of a prototype drug, but we still need to probably improve its properties. It's still, maybe it doesn't get absorbed by the human body very well. Maybe it's not quite as potent as we'd like, so the pill would have to be too big. We need to improve the properties, and to do that, one by one, chemicals are made, manufactured, handmade, one at a time, each one taking about a week or two to make, carefully crafted, adding little different variants onto those chemicals to try and make them fit. So if you look at the next one, the orange, look how beautifully that now fits in the pocket. That's going to be something that binds stronger. And so when we've made those variants and then test those variants, the game plan then is to overall to improve the properties. We're looking for efficacy, which is activity, selectivity, a reduction in what we don't want it to bind, which will then make it safer. And of course, we also need to pay attention to having something that is at the end of the day patentable, such that it can be protected so that a drug actually can move to the marketplace. So that's the kind of general game plan that we have been following. And I'm now going to pass over the baton to Doug, who's going to tell you about how we've used that process in a couple of advanced projects. These are no longer at the very beginning in target identification. These have now moved on to where we have prototype drugs and then moving to the clinic, which is super exciting.
Thank you, Michael. Finally, as Michael stated, you're going to hear from Dr. Douglas Sheffler, a research assistant professor in the Cancer, Molecules, and Structures program at Sanford Burnham Prebys, and has spent more than 15 years working on novel therapeutics for central nervous system disorders. Dr. Sheffler joined the Institute in 2012 and has a distinguished record of drug development that includes 8 issued patents and additional patent applications pending. His research team probes the interactions of drug candidates with proteins and cellular signaling pathways that play a role in neurological disorders and cancer, with a particular focus on therapeutics for drug addiction and major depression. Doug, please go ahead.
Thank you, Joe. So what makes a substance addictive? I know there's a lot of complications on this slide, but we're just going to keep it really simple. We've talked about these neurotransmitters that are how neurons communicate to one another. The neurotransmitter that really matters for addiction is dopamine. Dopamine is the chemical that makes you feel happy. It's rewarding. It's actually involved in what's known as the reward system in the brain. And there actually, as shown on the right in this slide, there are all these different pathways for the reward system. And none of the actual names of these are that important. The important thing is that the reward system is what its name applies. It rewards you. It's a survival mechanism for humans. If you do something that's good for you, you should be rewarded so that you do it again. So for example, we need to all eat. So when we eat, we sadly sometimes derive too much pleasure from it, which can lead to obesity, but it's still a natural reward. When people get the runner's high and they're actually running, again, it's the reward system. All these things stimulate reward. But what happens is drugs of abuse, all drugs of abuse can hijack this system. And it can make you feel the motivation to actually take that drug again. And if you keep using the same drug on repeated use, you actually change the way the brain actually responds to dopamine and how dopamine's handled. That is part of what makes you actually get addicted to a drug. Now, I know this slide's a little complicated, but we're just going to keep it really simple. To know if these drugs actually work, we have to be able to show that they work in lower species, for example, rodents, mice, or rats. So as it turns out, you can actually train rats or mice, and actually most organisms, to be addicted to any substance. And the two main procedures we use are called intravenous self-stimulation and cue-induced reinstatement. Self-stimulation is pretty simple. You can see an image of the rat over there. They have a little catheter that's implanted that delivers a drug straight to their brain. And they get this in a system where they're being trained. So they have two little levers. They have an active lever and they have an inactive lever. And they have a little nose cone where they can actually poke as well. And they have a tiny little light that's right by where they would get a reward. And what these animals are being done, they're actually being trained to press an active lever a certain number of times to get a reward. In this case, an infusion of nicotine, or an infusion of an opiate, or an infusion of cocaine. All drugs of abuse can be actually trained animals like this. And they will actually actively press that active lever to get as much reward as they can. For example, if you train animals to do cocaine, they'll actually just sit there and hit that button as fast as they possibly can to get as much cocaine as they want. So you train them to do this, and then after you train them, you can look at how your novel medicine for this particular disorder can interfere with that. And this is a drug-seeking behavior. So for example, does the medicine you're giving the animal cause them to actually seek the drug less? That's one aspect. Another aspect is that, as many of you have heard, what is the big problem with drug abuse? You get addicted to a drug, you then try to get off the drug, and you have two major problems. First off, most drugs actually cause massive withdrawal, and the symptomology is quite horrible, so it's hard to get off the drug. And then when you do get off the drug, you then can have a situation known as relapse. So what relapse is, in this case, we actually call it reinstatement of a behavior in animals. Relapse is pretty simple. Let's say you're smoking cigarettes and you get addicted to smoking, and you've always smoked a cigarette with 2 or 3 of your friends on a particular, playground or a particular structure that you're smoking with them. What you're doing is you're actually training yourselves, much like Pavlov's dog, that you're going to respond to a certain set of environmental cues. And those cues can be lights, sounds, friends, environments. Any of these things can trigger your brain that you now think, oh, I should smoke again. Like you finally get off of cigarettes and then you realize, oh, you're hanging around with the same buddies. Okay, I should smoke. And it's very hard to overcome this, which is why a lot of people relapse to drug taking. You can do the exact same thing with mice and rats. You train them to actually administer the drug, just like I just described, and then you actually stop giving them rewards. And they'll eventually teeter back down to not taking it. So they've essentially become abstinent, just like a human would. But remember I mentioned there was a little light that's actually going on every time they get a reward. Well, we're training them with that environmental cue that when they see that light, they think they're getting a drug. So if you actually go for time, have animals back at a stable baseline where they're not responding on either the active lever or the inactive lever, and all of a sudden you put that light back on, the animals thinks, oh, I can get drug again. And they will start hitting and actually reinstate, which is essentially relapse to taking the drug. So you could also look at how novel medications might interfere with that process to see whether or not they might work with relapse. So let's go back to an image that was actually shown a little bit earlier. And these are neurons with a synaptic cleft in between. We're going to zoom in a little bit. In this case, the neurotransmitter is actually glutamate. So one of the things that we work on, in particular Nick and I work on, are these receptors called metabotropic glutamate receptors. These are part of a large family of receptors in the human body that respond to the neurotransmitter glutamate. There are a lot of different types of these receptors, but let's just focus on the two we're really talking about right now, which happen to be subtypes 2 and 3. And these receptors, when you activate them, what they can actually do is they can change how dopamine functions in the brain. And it's not really an on or off response. Think of it more like a light dimmer switch, so that you can actually move the rheostat up or down, and you're just slightly tweaking how these cells actually can respond to dopamine. So what you can actually do is you can do these sort of animal models. So SA is self-administration. That's sort of measuring drug seeking, as I mentioned earlier. And the gray bar is that these animals actually will infuse, in this case, nicotine. They're trained to do that. And then you can administer our mGlu23 activator, in this case, which is this compound 801315. And what you see is the bar goes down. So when we give the animals these drugs, they don't actually seek or want to take the drug of abuse as much, in this case, nicotine. If you move over to this reinstatement, which is the relapse protocol, these animals are trained up in nicotine. They actually get back down to baseline and what's called extinguish the behavior so that they don't respond anymore. And again, that little light cue is there. And then you bring back the light cue. And these animals are actually not actually getting nicotine at this point, but they think they're getting nicotine, but they're trying to respond to get it. So they actually respond a lot to try to get it. And again, this compound that activates mGlu2 and 3 blocks that. So it actually indicates that at least in rodent models, the drug might help people with relapse. In addition, we have a lot of other data. I'm just showing some cocaine data here as well. So this is actually relapse to cocaine use. Again, the drug works quite well. So going back to our drug discovery process tree, where's this at? So we mentioned earlier on that most academic work focuses on this light blue area. Well, this particular work is something that Nick and I have been working on for well over 10 years. It's actually close to 16 years at this point. And we are very, very close to filing an investigational new drug application with the Food and Drug Administration. And we will likely be in man in phase 1 human clinical trials spring next year. Now, for the second program I want to talk to you about, we're going back to our same schematic. This time we're talking about a slightly different neurotransmitter. It's known as neurotensin. But it's the same general idea. You have these neurotensin receptors, which are called NTSR1. If you activate them, it's the same thing. It actually can act to modulate, like a dimmer switch, how dopamine functions in the brain. So what our group, including Michael Jackson and individuals at the screening centers have worked, they've actually worked out these what are called biased modulators. So I don't want to get into too many details because it gets too complicated, but any receptor in the body, it's not just one thing that they do. They do hundreds of different things. And there's all these different types of signaling cascades that go on. Well, you can actually design molecules that only do one or another of those pathways. So in this case, they've designed these drugs that actually are biased. They're actually biased toward this pathway known as the beta-arrestin pathway. And none of that really matters other than the fact that it's a pathway that we think, based upon all the preclinical research, would be good to help people with addiction. So what Michael's group has done is that they've developed these drugs. They work extremely well in those cocaine models that I mentioned a few slides ago. So they actually have a very, very profound effect on blocking cocaine seeking. So this represents a completely new class of drugs, and it's something that's going to take a lot of work to get to, but we're still in late stage optimization of this program. And they actually hope to be in phase 1 clinical trials, likely in 2023. So finally, what I wanted to do is actually mention a few acknowledgements. First off, we can't do any of this work without the support of the National Institutes of Health, in particular the National Institute on Drug Abuse, NIDA. These projects are all funded through NIDA. We would not be anywhere without NIDA. It's absolutely necessary to have this sort of work. These are, as Nick mentioned, as Michael mentioned, these are very expensive studies, and the National Institute of Drug Abuse is putting up tens of millions of dollars to help us get these into people, to try to help out addicts. In addition, philanthropy is highly, highly necessary to move these projects along. And I list down below the actual funding sources and the groups that collaborate with the mGlu2 3 Activators Project, which I'm heavily involved in, as is Nick. We also work with UC San Diego and a small biotech known as Camino Pharma. And on the NTSR1 side, that is driven through Michael Jackson and Anthony Pinkerton at SBP, and through collaborations at Duke University. And with that, I will turn it back over to Joe.
Doug, thank you so much. We're now available to take questions from the audience. I would remind you to use the Q&A button on the bottom of your screen, and we will filter the questions to our scientists. One of the things, Doug, I wanted to ask you, because you and I had discussed some of this before, previous medicines had acted more as an on-off switch for the dopamine, whereas ours is a dimmer switch. And you would explain to me that there were some serious side effects if you just turn off the dopamine, and that's why we think this is a better solution for that problem. Can you take us through that real quick?
Yeah, so it's highly convoluted, but essentially you have a reward system in the brain, and that reward system responds to any reward, both natural rewards, which we consider food, you need to sleep, things along those lines that are naturally rewarding and actually generally provide happiness. But in addition, they also are hijacked by illicit drugs of use. you're trying to function in a way that you're predominantly or completely impacting the unnatural rewards, the drugs of abuse, but not touching the things that you need to do. For example, if you had someone addicted to cocaine and you got them off of cocaine, but they stopped eating, that really wouldn't work so well. So that's really the gist of it.
Okay, I just want to remind people again to go ahead and send in questions. So that was another question I had, though. We can turn down the dopamine receptors for the drugs, but we can leave the runner's high that you're still going to get, or the working out or reading books or whatever other reward system that we get from the brain.
And that's the idea. So like, for example, let's go back to Chantix, which actually works on the exact same receptor system that nicotine works on. With that drug, you're actually preventing nicotine from working in the body. Well, you still need nicotinic acetylcholine receptors to actually function in the brain. So that's why that drug has a lot of side effects, and for a time had a black box warning from the FDA. The drugs we're working on don't have direct action. They're all indirect modulators of the dopamine system. And that allows you a lot more control over trying to just modulate it back down to a normal level, as opposed to just saying, I'm shutting dopamine down or I'm going to overactivate dopamine. We're just trying to tweak it to get it to where we want it to be to help people.
Nick or Michael, I would pose this question to you. The existing drugs, Nick, that I guess you talked about, the bupropion and things like that. How do those drugs work right now?
Yeah, that's a great question, John. Following up what Doug was saying, one of the issues with smoking is that the current treatments primarily all act on the same target as nicotine. So, for example, obviously smoking, nicotine is being delivered to you through a cigarette, absorbed in the so the nicotine is absorbed in the lungs and then gets to the brain. So the treatments for that include things like nicotine gum or nicotine patches or Chantix, which is yet another. It's a drug that mimics the actions of nicotine. So, you know, this is really it's not really a huge advance. Whereas the approaches we're taking. So on the one hand, we're modulating glutamate. On the other hand, we're modulating neurotensin. So these are very novel, different approaches, which we believe are much more powerful, potentially more powerful, as an approach to treat these problems.
Michael, let me throw this one to you. Are these drugs that are in development, are they addressing, the question here is craving, which often drives relapse?
We would anticipate so, for sure. You know, that's what they were designed to do, and as Doug sort of showed you, the animal model that we have is set to try and mimic that, equate that situation. Now, obviously, the biology of a rat and a mouse isn't quite the same. They're not maybe quite the same as a human, but it is remarkable that these animal models that we have in our hand do seem to mimic many of the aspects that you unfortunately see in patients. So yes, we anticipate they would do. And I think just going back to the point that Nick made, that, you know, there hasn't really been a new drug for addiction approved for a long time. And even those drugs that are approved are really not really what I would call smart bombs particularly, right? They're not, you know, today, don't forget, many of those drugs, if they came out in 2005, they first were started work on in 1990, right? It takes 15 years to come up with a drug, right? We're working now, it's now 2020, we're working with 30 years more insight into biology and the brain is the final frontier. It's the most complicated part of the human body. And we're getting there, and we have to develop these tools, and we're going to learn, right? But we can't sit there and not do anything, right? Which is, I think, being the frustration. And it is really exciting to be coming up with completely new medications that have never been anywhere close to this sort of thing before. And they have very exciting profiles in our animal models. And that gives us a great deal of hope.
Great. Thank you very much. Doug, I'll pose this one to you. Do we anticipate that these new medicines that we're coming up with, are they going to require lifetime use, or will they result in changes that the person can eventually get off the medications and stay off the drugs?
It's hard to tell for sure, but the hope is that it would be a drug similar to like what you try to do with nicotine replacement, that you have the person take the drug while they're trying to quit smoking, for example. It helps them have less cravings, so that helps them to get off of the cigarettes. And then it helps them and prevent them from relapse. Now, if you can get someone past a certain point, past relapse, and hopefully help the brain start to rewire itself to a non-addicted state, if that can even happen, it would not be something I would think you would need to be on forever. It's just, it depends. We're not going to know until we get into higher organisms.
Okay. There's a question here that if it works for cocaine, would it work also for meth? Are we working on something for meth too? And I guess the larger question is, will we need to develop different medicines for different drugs of abuse?
It depends on the exact receptor you're going after. What I can tell you about the metabotropic glutamate receptors in particular, we have a lot of data that it should work for nicotine. It should work with opiates, including heroin. It should work with cocaine, and it should work with methamphetamine. Now, there's other drugs of abuse out there, like, amphetamine and methamphetamine sound the same, but they actually have very different ways that they work in the brain. So while it could theoretically work for meth, amphetamine is a little more iffy. And for the neurotensin receptors, based upon the mechanism of action, there are a lot of drugs of abuse that could theoretically work for, and methamphetamine would be one of them. Now, it's what you typically do, at least on an academic level, you try to focus on one drug, because the reality is you have to get the drug up to the point where it's tested through phase 1 clinical trials in man. Once you do that and the drug is deemed safe, you try to get what is essentially whatever the lowest bar is to try to get it on the market. In our case, it happens to be tobacco. And in Michael's case, it may very well be cocaine addiction. But once you get that drug on the market, then you back up and you go through what the higher bars are, because running clinical trials for meth patients and heroin using patients is actually a much more difficult prospect than running a tobacco clinical trial.
Okay. Nick, let me pose this question to you. Would it be expected that these drugs need to be used with behavior modification therapies, or would they work sufficiently by themselves?
Yeah, that's a great question. And the most likely way would be to include therapy, some form of therapy and support. One of the interesting things about smoking in particular, that it usually takes several goes round for a smoker to quit. So they'll quit for a while, and then something will trigger this, what we call reinstatement, and they go back in. So combining the drug with some form of supportive behavioral therapy makes total sense for the long term.
Okay. Michael, I want to go to a question that we talked about earlier today, which is pharmaceutical companies and their perhaps maybe reluctance to step into this space. Now, we clearly seem to have a partnership with one pharmaceutical company. What's preventing them from being more active in this space, and instead that it's come sort of to academic medicine to fill that void?
I think it's the risk, right? I mean, they're for-profit organization. I mean, I used to work for Johnson & Johnson. I've been in that world. I have sat in committees where people sit around and agonize over where to spend essentially the shareholders' money, right? I mean, they're looking for a return on investment. And so it happens to be the case that actually drugs for neuropsychiatric disorders and neurodegenerative disorders, both of them, are both areas that they have experienced a lot of challenges with in clinical trials. And part of that is the very high placebo effect. All right. So it takes very large clinical trials to see activity, that activity is not so easy to pull out and to extract. And there are other areas that they could place their research, their drug discovery and development dollars. And it's a for-profit organization, right? So what I would tell you though is that no one of them would in any way challenge people to say there isn't an unmet medical need. The unmet medical need is intense, right? And so, why aren't we doing something about it? Well, like I said, I think we have been encouraged into this space to do something about it by the government. And yeah, I think it is the hope that should we de-risk these projects sufficiently to show that there is a clear path forward in the clinic, then the pharmaceutical companies can be encouraged to come back and come and join in and help, because we would need their help, frankly, towards the end game. They are the ones with the experience to do these very large trials. But that is, I think, why I don't, it's just business,
If the pharmaceutical companies didn't jump in, would NIDA be able to fund clinical trials?
They would and do, but I think they too would prefer to partner also. This is not a, pharmaceutical companies are ethical companies. They too are trying to help patients and use their capabilities to move things forward. But I do think that, they look at this and, recognize that they could use their dollars somewhere else to create greater value for their shareholders. And that's just how it is.
So based on where we are right now, what are the next steps? What do we need to go through to get from where we are now to get into these clinical trials?
Well, I think maybe Nick could answer that best for the mGlu-R compound. For the neurotensin compound, well, that's in the late stages of what we call clinical candidate selection, where we are trying to identify the appropriate formulation that generates the right exposure of the compound across multiple species. Once that is set, we actually have an agreement with NIDA that would trigger a next step in the development process. Nick, maybe you want to just say the exciting stage that you're at currently with the mGlu-R compound.
Sure. So for our mGlu2 positive modulator, we've completed what we call the preclinical IND enabling studies. So it's ready for the clinic. What we're doing now is we're compiling every single scrap of information we have on the drug and putting it into a huge document, everything related to its potency, efficacy, safety across species, its manufacture, and so on and so forth. This document, this huge document, will be ready by December, and then we'll submit it to the Food and Drug Administration. We have a meeting with them in mid-February when they will ask us questions, we will ask questions of them. After that, essentially they will approve us moving into human clinical trials, which we have scheduled for May of 2021. So that's where we are with that.
Michael, back to you for a second. We have a question about when drugs come to market. Clearly, a pharmaceutical company is going to make their profit on it. If a drug comes to market, does Sanford Burnham Prebys get a piece? Does money go back to the NIH? Do they reinvest that in research?
There will be components to do with the originator of the drug, the people who discovered it. Ordinarily would be recognized. So Sanford Burnham Prebys would be recognized for its contribution. But I think you need to also realize that it's going to take a lot of resources to get it all the way through to the end game. I think, as Nick mentioned, some of these clinical trials can be $50 to $100 million, or more, depending on. And so, the people who put that type of money up front will also be expected to be rewarded for the risk they're taking. So, it's at the end of the day, though, we are just proud to be contributing fundamentally towards medicines of the future for diseases that really are true on the medical needs. These are devastating diseases, and we're doing our bit. Obviously, we would anticipate eventually being recognized for some of that contribution and value creation. But that's not our drive right now. Our drive is the science. Our drive is the patient.
Okay. I think we have time for, let's say, 2 more questions. One question here, Doug, I'll throw this to you, or perhaps Nick. Do you anticipate that the drug may likely make an addict far less sick as they are going through the process of getting off the substance, which then also would then lead them, it might be more likely to stick to the rehabilitation efforts?
That's tricky. So in particular, that would be whether or not it actually ameliorates withdrawal symptomology. And there's some drugs that it looks like an mGlu2 PAM would actually help with a little bit, but it's really hard to know whether or not that's something that's actually going to work in people. And that's actually fairly hard to model in animal models, unfortunately. Like when I say animal models, I mean rat and mouse. Because that's how well can you ask a mouse or rat, how are you feeling today? That's why it actually gets hard to model those sort of things. You have to actually get to the point where you test it in people.
Okay. And the last question, and Doug, I would probably go back to you. Someone asked if we could please reiterate how the modulation works and how this indirect method compares to the direct method.
Got it. Actually, it's really complicated, but we'll just try to keep it as simple as possible. So there are a lot of different neurons of different types in the brain. And Nick mentioned at the front end that you have all these different brain regions. So let's take, for example, the extended amygdala has a structure in it known as the nucleus accumbens. It also has what's called the ventral tegmental area. There are neurons in that area that are not dopamine releasing neurons. So there's different types of neurons in the brain. Some just release one neurotransmitter, some release lots. So let's say there's a dopamine releasing neuron. Well, there's also these other glutamate releasing neurons that are coming down to synapse on that dopamine releasing neuron. Well, that glutamate neuron activates this neuron. Well, if you actually activate the receptor that's on this neuron, it actually makes this whole system activate this neuron less. When you activate this neuron less, this neuron releases less dopamine. So what you're doing is you're kind of changing the whole dynamic around the synapse so that the end result is less dopamine. So you're still getting the dopaminergic response and that dopamine is still being released, but you're kind of bring it back down a level where it's hopefully more normal. And the way different drugs of abuse release dopamine varies wildly. So, for example, methamphetamine actually causes a massive dopamine dump. Cocaine blocks dopamine reuptake, which means that dopamine sticks around longer, so the concentration goes up. They're all different, but it's all dopamine. So you're just trying to just come at this dopamine signaling in different ways to tweak it to get it back to normal.
Got it.
It's way more complicated than that, but that's the short of it.
That's why you guys are the scientists doing the work, and we're just listening. Thank you, all of you. Thank you, gentlemen, and thank all of you for joining us today. If you or anyone you know needs help with addiction, please, you can reach out to the hotline on the screen. We also hope that you will join us for future programs highlighting other areas of our research. Thank you again for attending. And if you would like to learn more about or contribute to any of our research, please contact me at either the email or phone number that will appear on your screen. Thank you, everybody. Again, we look forward to seeing you next time.